you guys like interact and put your comments in the chat box so that I'm able to interact with you and make sure that you are understanding everything during the whole session. So with that, I would like to begin the session here. So today is the day one, which is going to be the building block, uh, what makes us and what breaks us. So this is all about DNA. So let's start. So to give you an overview, what we are going to look in this presentation today is molecular basis of life. So the basics of the DNA, the composition of the DNA. Then we are going to have a look at the structure of the DNA. Then we will briefly discuss what is the function of the DNA, that is what is a gene. Um, what is a gene will also be discussed in the further sessions. So I'm just going to discuss this in brief. Then we will learn something about central dogma of life, and I will then summarize the sessions. So I'm going to teach you very practically so that you can apply this knowledge whenever you look at bioinformatics data. All right. So the DNA, as we all know, has been defined or abbreviated as deoxyribose nucleic acid. So it is the foundation behind our existence, and it has been long existing before we all existed as humans. So it is a very preliminary biomolecule, and this particular biochemical component is present in almost all organisms. There are exceptions such as viruses, but almost all organisms have their genetic component as the deoxyribose nucleic acid, aka DNA. All right, so let's discuss in brief what is DNA. So DNA is a double-stranded molecule that is twisted into a helix. So I believe in simplicity. So I don't want to give you a list of complex jargons of biology to make the biology complicated, because as it is more complicated to begin with. So this is the structure of DNA, and you do not need to worry about the technical details of it, because I'm going to go through whichever technical details are needed for you to make your journey in bioinformatics smoother. So just have a look at this structure and how it looks. And DNA sometimes is also defined as polynucleotide. So whenever you see this word, do not get confused as to why polynucleotide is used, because one component or individual units of the DNA are called as nucleotides, which is why many nucleotides coming together is a polynucleotide, which essentially means DNA. It has been defined as a complex biochemical entity uh, in biochemistry, which carries the information which is needed to build an organism. So this is basically like a dictionary that a cell refers to in order to build an organism. Now, some of the basic components which are present in DNA are: first, the nitrogenous base, the sugar, and the ion of the phosphoric acid. So do not worry about these terms, because we are going to discuss them individually. All right, so let's have a look at the nitrogenous bases. So the nitrogenous bases in DNA are A, T, G, and C. Um, and I will let you know about what the A, T, G, and C means, because I want to make sure that we are running through all the basics. RNA has A, U, G, and C. So instead of T, the RNA has U. And there are two types of nitrogenous bases, that is the purines and the pyrimidines. So this is just for you to understand that there are two types of nitrogenous bases. You will not encounter the use of word purine and pyrimidine otherwise inside bioinformatics too much, but just so you know that these particular chemical components are differing in their structure, and hence the chemical properties will differ a bit. And to show you how they individually look, this is adenine, which is A, this is guanine, which is G, this is cytosine, which is C, this is uracil, which is part of the RNA, not part of the DNA, and this is thymine. So Just observe that the adenine and guanine have this double ring structure, and the cytosine, uracil, and thymine have this single ring structure. So I hope that this part of the biochemistry is clear for everyone. What I want to remind you is this is just so that you know, and we all have brushed through the basics. You do not need to, by heart this. Just remember this key point, that the DNA has A, T, G, and C, and RNA has A, U, G, and C. All right. Now let's discuss about base pairing. So I know that a lot of people who have enrolled today for the class have come from different backgrounds. So bachelors, masters, some of you I saw have also been working as pathologists. So I guess you all know about the basics, but I want to make sure that you know the basis of the wet lab and how these properties of the DNA are being used or exploited. So about the base pairing, so adenine, that is A, pairs with thymine or with uracil. So in case of the DNA, adenine or A pairs with thymine, and in case of the RNA, it pairs with uracil. So A and T come together, and G and C come together. So A and T would be this, for example, and G and C is this here. Now you need to remember that this particular bonding which you see here, this is a bond, this is a bond, and this is a bond as well. So this is through hydrogen bonding. Okay, and also in case of the A and T, they pair through hydrogen bonding. Now I want to discuss why this particular property is important. So just hold this thought that this particular bonding is through hydrogen bonding. Now, why this is important, you will encounter all of it in the upcoming sessions. So just hold this thought that the bonding is via the hydrogen bonding between the base pairs. Now let's discuss about sugars. So there are two types of sugars. One is the ribose sugar and the other is deoxyribose. Now ribose sugar is this particular component. You do not need to remember about the structure of this particular sugar. What you need to remember, however, is that this is known as ribose because this has the hydroxyl group, and because this particular sugar does not have the O present, it is known as deoxyribose. That is, some component called oxygen here is missing, which is why this is known as deoxyribose. And if you remember, DNA is called deoxyribose nucleic acid. So you might have guessed that the DNA contains this particular sugar. Now let's discuss about the nitrogen base and sugar and how they come together. So now the nitrogenous base linkage to the sugar occurs via a bond called as beta N glycosidic bond. So you can see this bond here. Essentially these two are like the bonds are the same, but here it is in case of one type of sugar, and here it is in case of the pyrimidine, so the other type of sugar. So this is what you have to remember, that the bond is N glycosidic bond. This is important to remember because when we exploit the properties for any biological analysis using sophisticated machines, these particular chemical properties are exploited there. Now let's discuss about phosphate ion and linkage to sugar. So there are three serially bonded phosphate groups. So phosphate group is PO4 3 minus. So this is one phosphate group here. This is the second phosphate group, and this is the third phosphate group. So now since this is the first phosphate group which is attached directly to the ring here, it is known as alpha phosphate group. This is beta and this is gamma. So as you go farther away, you are going to label it as alpha, beta, or gamma. So what is important thing to remember here is that this particular bonding with the sugar of the phosphate bond happens via phosphoester bond. Okay, so this particular bond which is present between the phosphate sugar and the phosphate ion and the adenosine is via phosphoester bond. Now hold this thought, this particular thought, because you will also need to exploit this when you use biochemical reactions in order to get pure components of DNA. So yeah, you should remember this particular point when we discuss things ahead. Now, before we move ahead, I would like to make sure if things are clear for everyone here. All right. So I guess till this part it is clear for everyone. Now I would like to move ahead, and just remember you should hold this thought whatever we learned till now. So now I would like to discuss an important component, that is directionality of DNA. What do you mean by parallel and anti-parallel strands? So you might have heard about it or you might not have heard about it. Anyway, I'm going to discuss this because it is very important and critical detail. So if you look at the structure here, just ignore the information on the left hand side which is written. Just have a look at this structure now. So if you look at this particular end on the top, this is the phosphate ion, all right, as we learned in the earlier slide. And this particular phosphate bond is present, and this particular end is known as 5' end. So the part of the DNA which has the phosphate ion will be termed as the 5' end, and the hydroxyl group which is present on the other part will be termed as the 3' end. So now I will come here. So important thing to remember here is that 5' end is the phosphate group and 3' end is the hydroxyl group. Now, why is this important to remember? Because in bioinformatics, whenever you receive the sequencing data, you will never receive it randomly. So the sequencing, no matter however it is done inside a machine, when you get it out, the sequence will always be represented from 5' end to 3' end. So just have a look here. So while writing a DNA sequence it is always written in 5' to 3' direction, which means that if you happen to sequence a DNA or the anti-parallel strand, so the parallel strand means if you read the DNA sequence from 5' to 3' end, if you read the DNA from 3' to 5' end, it is essentially the opposite strand which is bonding with the DNA, because DNA, as we discussed, is a double helix. So the complementary DNA sequence is the 3' to 5' end. So that is the anti-parallel strand. So just remember that sequencing data will always be represented to you in 5' to 3' end. So just keep this in mind. All right. So now let's have a look at the structure of DNA. So what we see is that DNA diameter is around 20 angstroms. There are some components which are known about DNA, but since they are not part of what will be essential in bioinformatics for us, I'm not going to discuss what is major groove, what is minor groove, but I just want you to have a look at this particular structure of DNA. So sharing some facts with you, DNA is a double helix, and the original discovery was made by Watson and Crick using the data or X-ray crystallography image, which was originally derived by Rosalind Franklin in 1953. It contains anti-parallel strands, which means both the strands are complementary to each other. So if one of these strands, just follow my laser now. So if this particular component is represented from 5' to 3' direction, the dark gray strand, which is the second strand, will always be 3' to 5' end. So you should always remember that the DNA strands double helix is going to be complementary to each other, and this is precisely the property which is also being used. So if you have information about one strand of the DNA, you do not need to then have the another strand. So you can easily define what can the other strand be, because you know that they are complementary to each other, and what are the base pairing which is occurring. That is, A pairs with T via double bond, and G pairs with C via triple hydrogen bond. So as I discussed, the complementary base pairing occurs for A with T and G with C. And the important thing we discussed in the previous slide is that they have sugar phosphate backbone. Now, I would like to discuss one more important component here, is that you should remember that the sugar phosphate backbone is present. Now, I would like to ask question to the participants here. Can anyone define what would be the charge on DNA if I am telling you that we have the phosphate backbone? Yes, Shobhit, that is right. Sudarshana, that is right. Others, feel free to respond. There is no such question as a silly question. It is very important to be interactive. Akshat, that is also right. So I guess you have all understood the basics, and you are absolutely right. Uh, Before moving ahead, there's one more important question for you. So guys, we discussed that we have the nitrogen bases inside DNA, right? So A, T, G, and C, or uracil in case of RNA. Now, one more question to you all is, what would be the nature of DNA? Is it going to be acidic? Is it going to be neutral? Is it going to be basic? Okay. Can others respond as well? Okay. Okay. All right. All right, so you guys are right. And if other people have not understood by now, it is acidic because the phosphate group that we discussed, that forms the backbone, it can easily release the protons. Protons means the H plus ions. This is like basic chemistry. I hope you all are aware about it. But any component which releases the protons in a solution will be termed as acidic. So this is the reason why it is going to be acidic. Why I also discussed that we have the nitrogenous bases is because I wanted to see if the concepts are clear. So you might think that since the nitrogenous bases are present, it is going to be basic in nature. But always remember that, you know, whichever component is present and protruding outside is going to decide the charge. So the bases, nitrogenous bases, are facing inward, so they are not going to be able to decide the charge, sorry, the basicity or acidity of the DNA molecule. So the phosphate groups are outside, so they will be having the H plus ions with them or release the H plus ions, which is why only the phosphate molecules will decide the basicity or acidity of the biochemical molecule, that is DNA here. So just remember this as well. So why this information is important will be discussed in the next coming upcoming days. But to tell you, you know, when we want to derive a particular molecule from a solution or from a biological sample, we need to separate it. And in order to separate it, we need to essentially make sure that we apply the opposite charge or opposite um, basically pH to the solution or to the component of the tissue to separate the DNA. Because if you have the same component which is compatible with DNA, the DNA is not going to separate. So this is precisely the property which is used in order to extract a DNA from a tissue or a liquid solution. So this is why knowing the basics of the DNA are very important. Now I would like to move on to one of the important components which is going to be useful to all of you throughout biology and throughout bioinformatics. So let's discuss about the central dogma of life. So the central dogma of life says that the DNA gets replicated, and this particular process happens via the use of DNA polymerase. So replication essentially means making its own copy. So DNA polymerase is the enzyme which is used. Now the DNA then gets transcribed. The process is called as transcription, which means the DNA is converted to RNA, and this happens using the enzyme RNA polymerase. So essentially there are different types of RNA, but you can roughly say that the component which gives rise to a protein here, the part of the RNA which we are talking about here, is mRNA or the messenger RNA. So RNA polymerase participates in order to transcribe the DNA to RNA. And then um, when an RNA is read by the ribosomes, we get something called as amino acid chain. And if it is long enough and complex enough with all the foldings, we call it a protein. So RNA is converted to protein. So this particular component or this particular process is called as central dogma of life. So DNA to mRNA to protein. So this applies to almost all organisms except some viruses. Now, I would like to hear from the audience. Does anyone know of any virus which actually does not use DNA as the genetic component? So I'm also looking at the people who are watching live here. So everyone can answer. I want it to be interactive, so I am making sure that nobody's sleeping here and you are gaining most of what I'm teaching. Yeah. Um, so let me have a quick look. All right. So, yeah, coronavirus is right. Sudarshana, Shobit tobacco mosaic virus. Yeah, that is right. HIV, Sudarshana, that is right. Influenza virus, also that is right. Um, yeah, so tobacco mosaic virus is a plant virus. Talking about animal virus, what we have similar to it is rhabdovirus. Influenza virus is also an RNA virus. It is a segmented RNA virus. HIV is very famous, or let's call it infamous virus, which is also using RNA. All right. So thank you for answering everyone. Also, thank you for answering all the live audiences. I'm happy that you all are interacting. All right. So some viruses can reverse transcribe, meaning that they have the reverse transcriptase enzyme. All right. So mRNA is used in order to get to the DNA. So these are all the basics that I wanted to teach you today. And yeah, these basics will be applied to the bioinformatic data, and stay tuned because these are some of the most basic concepts and they look very easy, but all of us have the tendency to learn most advanced stuff but forget about the basic stuff. So it is going to be useful to you all if you all remember all these important points. So to take away from today's session, hydrogen bonds dictate the strength of the DNA double strandedness. The base composition of DNA is very, very important. The nitrogenous bases make the DNA basic. This is very important. Phosphate group gives the DNA an overall negative charge, and DNA is the key molecule which ultimately dictates the protein molecule, that is amino acid composition. So I hope that everyone has understood what has been taught today. And today's session was just some basic session, but nonetheless, it is very important for you to understand all further sessions, which will be even keep on getting a bit, I would say, not complicated, but advanced. So yeah, if you have any queries, feel free to write in the chat box, and I would be happy to answer all your queries. So one thing I would want to remind you here is it is only possible for me to answer limited set of questions. So I would be only able to see questions and answer them from the people who have enrolled in this session. These classes will be live for free for 2 days. So if people are interested, they can go through them, and I'm sure that you will be gaining most and find it very informative. And if you like it, you can enroll, and then I would be able to also see your questions. Shobhit Raj, yes, DNA is acidic. That is right. Uh, Janvi, yes, I will be sharing the today's PowerPoint presentation with you as well. So people who are going live, I will also answer some of the questions you ask. Just give me a few moments, and I will address your questions as well. All right. Yes, Shobit, recorded lectures will also be there. So as soon as today's session ends, we will upload it on the drive, and this will be available as a private session, and it will be shared with you all. Whoever has enrolled for the course, you will get the offline recordings. Okay, so just hold on for a minute. I'm just seeing some questions from the live YouTube. Uh, how DNA decides that it converts into protein or translates genetic information? So, this is part of Akhtar Jameel. Okay, this is part of the process wherein DNA will get converted to RNA and then to a protein, because this is happening during the cell cycle. So it is not happening randomly. So during the cell cycle phase, when it is the synthesis phase, typically the chromosomes will get untangled, and during that particular time or that particular phase, the DNA will get the involvement of the enzymes called DNA polymerase or RNA polymerase, whichever is applicable, and then it will get replicated, and then there will be replication, or there will be also transcription which is happening. I hope that clears your question, Jamil. um, I would like to once again remind everyone who is attending today's session that the application link for this master class is still open, and you all can go and still enroll yourself for this master class. All the details are available in the description of this video. So there is one more question from someone asking, sir, why is DNA negatively charged? So, okay, all right, I will explain it once again. Um, so, I discussed that DNA has phosphate group present, okay? So DNA has this phosphate group, all right? So since it has phosphate group, you see here it has negatively charged O, that is O minus. But normally this will have OH here, H, okay? OH, OH. So whenever H is present here, it means that a molecule, when it is formed, when it is present in a solution, which is the typical component or part of the cell, it is always going to release this H which is present, okay? And any component or any biomolecule, when it releases the H plus ions in the solution, which means when the solution will be having excess of H plus ion concentration, it will be termed as acidic, which is why we say that the DNA is acidic in nature. So any biochemical moiety or component which has the ability to produce or release H plus ions will be termed as acidic, and this is precisely why DNA is an acidic molecule. Let me just have a quick look if anyone else wants to ask any question. All right. Just everyone who is, just a second. So myself, Divya, I have completed this. Okay, so Divya Anasa, if I spell it correctly. So Divya, what you need to essentially find out is what do you want to pursue your PhD in. So of course, this is like a topic which is like very broad, or which can be discussed in details. But to give you an answer briefly, you need to figure out what you want to do in your PhD, because there is no generalized PhD. So PhD is going to be very specific. So first step would be to figure out what do you want to do your PhD in, what topic actually intrigues you. So find out the places or the universities where the topic of your interest is available, and after that you should pursue that particular professor. Just write to this professor after reading what they have been publishing and what they have been working on, and then you can decide which university to go for. And I will still tell you this, that the university is not very important for a PhD. Like looking at the university, you don't need to go for Harvard or MIT or some elite class universities. Doing it from a good university is all right. You don't need to see the rank of the university, but seeing to it, making sure that you are doing your PhD in something that interests you and that will make you gain some significant knowledge is important. So yeah, Divya, you should first look for the topic of your interest, and then you should look at the university to choose for your PhD abroad. Akshat, what are the key areas I should focus on most for bioinformatics? So, we learned about the central dogma of life here, So, the easiest part to learn in bioinformatics right now, and also the most important part, is to start with programming. I would say that programming is most important to learn right now. So, Python programming, learning Python, learning R, learning Linux basics, and just so you asked, Biotechshalaa is also teaching all these topics as well. Um, and the other components will be first starting with the genomics, because that is how also central dogma starts. So if you know genomics, of course you can go for transcriptomics and eventually for proteomics. So just go with learning some key languages with which you can work with offline on your PC using Linux, and then focus on learning genomics. So I think that would be the right pathway, according to how I have always approached bioinformatics. So Sudarshana, sir, I'm willing to understand about industrial PhD abroad and in India. So Sudarshana, regarding the industrial PhD, you always need to see if the industrial PhD is funded, because such positions are limited. I can tell you that, for example, company like Boehringer Ingelheim uh, in Germany, they provide industrial PhD. They have their applications open, I think, twice a year, but since last year they have only started doing it once a year. So you have to apply very early, and you have to be an extraordinary candidate if you are applying internationally. Otherwise, companies like Cipla, companies like for example, Thermo Fisher, companies like Biomerics, they also provide international programs which are industrial based PhD. Shobhit, sir, what are the scope of bioinformatics in India as well abroad? So, Shobhit, good question. So, one of the things that one needs in order to learn bioinformatics is essentially only a laptop and server. And of course knowledge regarding bioinformatics. So in India, I would say, since we have been very good since past 15 years on the IT part, um, it's having a very good scope in India as far as professional career is concerned. Even if you were to consider academics, it is really growing and expanding in India as well as abroad. So yeah, it has a wide variety of applications as far as academia is concerned, or even industry is concerned. So it has good scope. Dr. Jamil from YouTube has asked one more question. So I want to ask question: how can RNAs from 5 prime to 3 prime or 3 prime to 5 prime, because RNA has single strand? Okay, Jamil, this part will be answered tomorrow, because tomorrow I will be taking the further part or further component of this session, wherein we will learn more about RNA as well. It will be in brief, but this is where you will also learn from it, and this is where I will tell you how it happens. Akshat, how bioinformatics can help us to understand evolution. So, Akshat, to answer your question very briefly, we have DNA sequences and we have the protein sequences. And if you have read about it, you should know that some of the proteins and DNA sequences are highly conserved, which means that right from a bacterial protein or bacterial nucleotide sequence to human nucleotide sequence or a human protein, the function of this particular biomolecule will be conserved, which means that there will be no change in evolutionary ladder. So these are the conserved proteins, and some of them will have a lot of changes. So gathering information about both, if we analyze these sequences, we can generate something called as phylogenetic tree. And a phylogenetic tree will help us to identify how, for example, a human is distant from a gorilla, and how both of them are extremely distant from some prokaryote like E. coli. So this is how we can understand evolution. So this was just a short example. There are many ways to do it. Typically, people think that considering the tree, phylogenetic tree on an amino acid level is much better than doing it on a nucleotide level, and there are differences in opinions there. But yeah, this is how basically bioinformatics can help understand evolution. Yes, Jamil, the class will be conducted from 8 to 9, as it was also conducted today. So I'll just give everyone a minute. If there are no questions, I will then hand over to Muskan to conclude the session.
Thank you, Dr. Siddhesh. So, before we end today's session, I would just like to sincerely thank you for such an insightful and engaging lecture. And on behalf of everyone at Biotechshalaa and all of our participants, thank you so much for taking the time to teach us today. We truly appreciate your efforts and the knowledge that you have shared with us today. And thank you to all the students for joining us today. We hope that you found today's session very valuable. So we'll see you tomorrow for day 2 and have a wonderful evening.
Thank you, Muskan. Thank you, everyone, for attending and being attentive.